In this video we will discuss about this CD8 plus T cell characterizations. So in the previous video we have understood that cluster populations perform well compared to the singlet T cells, and we have also seen that in the patient's samples also uh, we have find out that the tumor cells and the T cells are occurred in the clusters, and the singlet T cells are also present. So there is a heterotypic T cells populations are present within the patients. So now they wanted to characterize different kind of these T cells that are exist in this patient and what are their cell state. So what it is, they took the patient samples uh, and then label them with different fluorescence markers, and then sort them using the flow cytometers or flow sorter, that is fluorescence activated cell sorting, and then characterize them using the single cell RNA Seq and single cell T cell receptor Seq data. And once they get this data, they overlap this data based on the T cells single cells uh, single cell RNA Seq data of the T cells are available in the public, and they overlap those gene expressions with the gene expressions that is seen from the single cell RNA seq and single cell T cell receptor 6, and they are able to find out that the T cells are in the 14 different state. Out of these 14 different state, they are able to see that naïve T cells are more commonly found in the single T cells, whereas the proliferating T cells are more found in the cluster T cells, either with tumors or with APC. And they also check this TCR clones, and they're able to see that TCR clones are more found, more expressed in the case of in the clustered conditions. It is either with tumors or with the APC, and whereas the single T cells has the lowest amount of the TCR clones available. And in order to further validate this point, they did they characterized these different T cells. There is single T cells clustered with tumors, and T cells clustered with APC using gene expressions. And they are able to see that the T cells that are single T cells are more expressed in the case of more expressed towards this virus reactive, and which is the common state for the bystander T cells. Whereas the T cells that are clustered with the tumors or with the APC are more expressed. Tumors or APC are more expressed in the case of this tumor reactive. So they are more reactive towards the tumors, and which can indicates that these T cells which are in the clustered form with the tumors or APC are more prone to have a antigen specific response against tumor antigen specific response compared to the single T cells. And thereafter they also overlap the clustered T cells data, the T cells data that are available of the patient, and they are able to see that the overlapping is higher in the case where patients shows high response towards infiltrating lymphocyte therapy that can be seen in this panel A, whereas the overlapping is low in the case where patient experience less response towards tumor infiltrating lymphocytes therapy. It can be seen for this high patients that can be seen or divided into the three categories, that is top 30 clusters and the medium 30 clusters and the low 30 clusters. So top 30 clusters overlapping shows the high reactivity or high overlap for the patients who are reactive towards tumor infiltrating lymphocytes. But as soon as the overlapping goes down, the reactivities, the relationship also goes down. So in the lower case, where the overlapping is minimum, it shows that the non-reactive overlapping is minimum, or we can say that the single cells gene expressions are more overlapping to the patients. In that case, those patients are less reactive to the tumor infiltrating lymphocyte therapy. And for the medium case, we do not see any significant changes. So this all indicates that the clustered formations of the T cells with the tumors seems to be a positive indications towards high anti-tumor efficacies or high efficacies of the therapeutic approach that will be used in the patient. So we will further discuss further characterizations of the CD8 plus T cells in our subsequent videos, as we have discussed only half of the figures in this video. So that's all for this video. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. Thank you for your time.